everyone, welcome to Learning and Interaction with Miss Aisha Khadir. Today we will start to solve past paper of O-Level Biology 5090. So we are starting with year 2010, paper 1, variant 1, MCQs number 1 till 10, and this is the part 1 of this video. Please like and subscribe my YouTube channel for getting more useful videos regarding exams preparation. And click the bell icon here. Let's start. So this is MCQs number 1. Okay. The photomicrograph shows onion epidermis. This is the onion epidermis. And you know that epidermis is the first layer of plant's organ. This is epidermis, and epidermis is made up of many cells. So which term describe the onion epidermis? Cell, organ system and tissue. So onion epidermis is the tissue of the plant, tissue of the plant, because it is made up of many cells. Okay. Identical pieces of potato are placed in sucrose solution of different concentration. After 3 hours, the mass of each potato piece is measured. Which graph shows the result of this experiment? Okay, as you know that there are two types of solution, hypotonic and hypertonic. Hypotonic and hypertonic. Hypotonic contain less amount of solute, and here solute is the sugar, and hypertonic solution contain high amount of solute. So that's why, and here the solute is the sugar. Okay. So when you place any cell, let's suppose this is the cell. When you place this cell in hypotonic solution, so it's mean that water is present in high amount outside the cell and in less amount inside the cell. What will happen? You can observe the water movement inside the cell. You will observe the water movement inside the cell. Water from outside goes inside. This will cause increase in mass and volume of the cell. Cell become turgid. While if you place cell in hypertonic solution, so you will observe the opposite change. Now what will happen if you place cell in hypotonic solution? So what will happen? Water is present in low amount outside. In hypertonic, water is present in low amount outside. So from the cell, water move out by exoosmosis. So you can see here, and cell may experience shrinkage. You can see here, when you are increasing the sucrose solution, what will happen? Mass of potato after 3 hours. If you can observe here, which graph shows the result of this experiment? In A, they are saying that if you are increasing the sucrose solution, mass of potato decrease. If the sucrose concentration, you can observe here, let's suppose if the sucrose concentration is 1, so mass of potato is low, and then high, and after certain limit, mass of potato will start to decrease down. Okay. You can observe here at the sucrose concentration 1 and the sucrose concentration 2. Which one is the hypertonic? Obviously this 2 is the hypertonic. So in this 2, more water should move outside. More water should move outside. So in this case, mass of potato will decrease, not increase. I can explain here. Let's suppose if here is the 1 and here is the 2. So if we are considering that 2 is the more hypertonic than 1, so at this stage, more water should move outside of the cell. So how is this possible that the mass of potato increase, mass of potato should decrease? So this is the wrong option. Same here, if you are increasing the sucrose concentration, gradually you are increasing sucrose concentration, this graph showing that the mass of potato is increasing, and it is not possible. When you are increasing sucrose concentration, so your solution will become hypertonic, and in hypertonic solution, what will happen in hypertonic solution? Water move outside. When water move outside, there should be the decrease in the mass. There should be the decrease in the mass. And look at this C option. This is the right option, because when there is the low sucrose concentration, it means high water concentration. Low sucrose concentration means high water concentration. The solution is hypotonic. When the solution is hypotonic, what will happen? From outside, water move inside, so mass of potato will increase, will be at its higher stage. Then gradually, when you are increasing the sucrose concentration, what will happen? The solution is start to convert from hypotonic to hypertonic, hypotonic to hypertonic, and in this way, more and more water start to move outside and cause shrinkage of the cell. So mass of the cell will gradually decrease. So this is the right option. Option number C is the right option. And this is also not possible that at high concentration, at hypertonic concentration, how the mass will increase, how the mass will increase. If the water move outside, mass should decrease. So this is C is the right option. Okay. Which of these processes require energy from respiration? Okay. When we are discussing about the transport, there are two types of transport: active transport and passive transport. Active transport and passive transport. In passive transport, substance move down the concentration gradient. Down the concentration gradient means from high to low concentration. Okay. While in active transport, substance move from low to high concentration, and you can say against the concentration gradient. So in active transport, you need energy. You need energy for active transport. But you can observe here, diffusion and osmosis. These both are the types of passive transport. Diffusion and osmosis is the type of passive transport, and from movement of substances from their high to low concentration required no energy. So for passive transport, there will be the no requirement of energy. For passive transport, there will be no requirement of energy. No energy is required. No energy is required. So which option is the right option? For diffusion, you need energy? No. For osmosis, you need energy? No. So option number D is the right option. Okay. Fourth one, cubes of hard-boiled egg white are placed in test tube containing 5 cm cube of water. Other substances are added to each tube as shown in the chart. The tubes are left for 8 hours and then tested for amino acid. Okay. Egg white contain what? Egg white contain protein, and protein should convert into what? Into amino acid by the action of enzyme. Okay. Which tube shows that pepsin is an enzyme? So the question is, cubes of hard-boiled egg white are placed in test tube containing 5 cm cube of water. Other substances are added to each tube as they are mentioning here. In tube 1, what they are adding? They are adding pepsin, which is the enzyme, and this enzyme should convert what? This enzyme should convert protein into amino acid. Okay. So in first tube they are adding only pepsin. In second tube they are adding pepsin plus alkali. For third tube they are adding nothing. For fourth, pepsin plus acid. For fifth, boiled pepsin plus acid. Six, acid and seven, alkali. And these are the basically result. Okay, which tube shows that pepsin is an enzyme. Okay, when we are going to study enzymes, so there are two main factors that can affect the activity of enzyme. Number one factor is the pH. Okay, so enzymes need particular pH for their working, and pepsin is the enzyme which is present in stomach of the mammals, and for the action of pepsin we required acidic pH which is maintained by HCl, which is secreted from our gastric walls. So enzymes are pH sensitive and enzymes are also temperature sensitive. So for the working of enzymes, for the optimum working of enzyme, the optimum temperature is about 37 degrees Celsius, because this is the optimum temperature of our body. So you can observe here that if you are adding pepsin only, there will be the no effect, because protein is not converted into amino acid. No effect. When you are adding pepsin plus alkali, so result of test for amino acid is also negative, because pepsin should need acidic pH for its working. When you are adding nothing, obviously there will be the no result. But when you are adding pepsin plus acid, yes, large amount of amino acid will observe. Why? Because in this tube what will happen? Protein digest into amino acid, and in this tube is reflecting that pH affect the acid, and basically acidic pH affect the pepsin, and pepsin start to work. So that's why pepsin is the enzyme, because it is pH sensitive. Pepsin only work in acidic pH. So test tube number 4 showing that pepsin is the enzyme, because pepsin work in acidic pH and release large amount of amino acid by digestion of proteins. But what will happen if you adding acid but boiled pepsin? What you have done boiling with pepsin. So here you can observe that pepsin is the enzyme, so it is temperature sensitive. When you are going to boil this, so what will happen? It will denature. So basically test tube number 4 and 5 is showing the property of the enzyme, that enzymes is pH sensitive and enzyme is temperature sensitive. So this test tube number 4 and 5 is showing that pepsin is an enzyme, because pepsin work in acidic pH, and boiled pepsin could not work, because boiled pepsin denature due to rise in temperature. So option number C is the right option. Okay, which sign shows that a plant has been grown in a culture solution deficient in magnesium? When magnesium is not there, so what will happen? Basically magnesium is required for chlorophyll synthesis. So if there is no magnesium, what will happen? Purple leaves and poor root grows, no. Small leaves and thin stem, no. White upper leaves and normal lower leaves, no. Yellow stem and yellow leaves. This is the right option, because in absence of chlorophyll, what will happen? Leaves start to become yellow, and also stem. Okay. Now in MCQs number 6, we are starting MCQs number 6. The graph shows the rate of photosynthesis of a plant at increasing light intensities at two different carbon dioxide concentration. The temperature is kept constant. Okay. Rate of photosynthesis, this is light intensity, 0.03% carbon dioxide, 0.06% carbon dioxide. What may be the limiting rate of photosynthesis at X, Y and Z? Okay. You can observe here, when you are increasing the light intensity, till X, so what will happen? Rate of photosynthesis is increasing. But after this, when you are increasing the light intensity, there will be the no effect. So till X, What is the limiting factor? Light intensity is the limiting factor. What is the limiting factor? Basically till point X, light intensity is the limiting factor. Light intensity. But as you can observe here, at 0.03% carbon dioxide, the rate of photosynthesis also limited. But if you are increasing the carbon dioxide concentration, what will happen? The rate of photosynthesis enhance and then become constant. So at Z and Y, what is the limiting factor? Basically carbon dioxide is the limiting factor. So which option is the right option? Option number C is the right option, that till point X, light intensity is the limiting factor. Due to the light intensity, the rate of photosynthesis will become limited. But when you are increasing the carbon dioxide concentration from 0.03 to 0.06, what will happen? There will be the increase in rate of photosynthesis. But after this, it also become constant. So at point Y and Z, what is the limiting factor? Carbon dioxide is the limiting factor. Okay. The diagram shows a section through a leaf. Which cell have the function shown? Photosynthesis and transport. 1, 5, 2, 4, 3, 4, 4, 5, 2, 3, 1, 4, 2, 5, 2, 5. Okay. Let us starting the labeling of the plant's cell. Okay. These are the basically palisade cells. These are palisade cells, and palisade cells contain chlorophyll. So that's why palisade cells can perform photosynthesis. Option number 1. This is they are performing photosynthesis. Okay, and these are basically spongy mesophils. It also contains chlorophyll. You can observe here. It also contains chlorophyll. So that's why they can also perform photosynthesis. Now 2 is the phloem. Phloem could not perform photosynthesis. And these are the xylem. Xylem also could not perform photosynthesis because xylem is a dead cell and they don't have chlorophyll in them, so they could not perform photosynthesis. And these are basically epidermal cell, lower epidermis. It also devoid of chlorophyll. You can observe here that there is no chlorophyll present inside, so it could not perform photosynthesis. So only 1 and 5 could perform photosynthesis. 1 and 5 could perform photosynthesis, not 2, not 3 and not 4. So 1 and 5 perform photosynthesis, yes. And 2 and 3 transport, yes. Phloem transport what? Food, organic food. And xylem transport water and dissolved minerals. So option number A is the right option. That for the photosynthesis, palisade mesophils and spongy mesophils are responsible, and for the transport, your phloem and xylems are responsible. Okay. The diagram shows a section of the small intestine in which partially digested food is being pushed along. Okay. So you can observe here there are two types of muscles, longitudinal muscles and circular muscles. They are antagonistic muscles. Antagonistic muscles means they work in opposite fashion. When longitudinal muscles relax, what will happen with the circular muscles? It contract. And when it's contract, so it will relax. So they are basically antagonistic muscles. They are working together but in opposite fashion. Okay. So when we are discussing about circular muscles, when circular muscles contract, what will happen? When circular muscles contract, lumen of intestine decrease. And when circular muscles relax, what will happen? When circular muscles relax, lumen increase. So you can observe here To accommodate this partially digested food, lumen should increase in volume. Lumen should increase in volume. So that's why you can say here that the circular muscles at 1, at this position, circular muscles is going to relax in order to increase the diameter of lumen. In order to increase the diameter of lumen. And if the circular muscles relax here, what happened with the longitudinal muscle? Because in question you have to describe the state of longitudinal muscles at 1 and 2. If at point 1 circular muscles relax in order to increase the size of the lumen, so what will happen with the longitudinal muscles? Longitudinal muscles will contract. Longitudinal muscles will contract at position 1. So longitudinal muscles is contract. And at point 2 what will happen? When you in order to move the food in further direction, this is the direction of the food. So when you are moving the food, just before the part of the intestine, just before the food start to contract in order to apply pushing force on food particle. So what is contracting? Basically circular muscles are contracting. You can observe here the lumen of the intestine is decreased, so circular muscles contracting. When circular muscles is contracting at this point, what happens with the longitudinal muscles? Circular muscles contracting. So longitudinal muscles will relax. So option number B is the right option, that at point 1 longitudinal muscles contract because circular muscles relax, and at point 2 longitudinal muscles relax because circular muscles contract. Okay, so option number B is the right option. In which region of the alimentary canal does amylase act? What is amylase? Amylase is the enzyme. And what is the substrate for amylase? The substrate for amylase is the carbohydrate. So amylase, the role of amylase is to convert starch into maltose. Okay, so where amylase act? First of all, amylase is secreted from the salivary glands. So due to the presence of amylase, what will happen? Due to the presence of amylase, the digestion of carbohydrate start in our mouth. So amylase work in the mouth, yes, and in stomach amylase become denatured because of the acidic pH of the stomach, because amylase work in alkaline pH, not in acidic pH. So in stomach amylase could not work. So option number C and D is the wrong option. Pancreas is not associated with the digestive tract. This is basically gland which secretes some juices but not the part of the GI tract. So this option is the wrong option, while mouth cavity and ileum is the right option, because first from amylase which is secreted from the salivary gland acts in mouth, then in ileum. When pancreatic juice arrive in duodenum, which is the first part of the small intestine, so from duodenum, jejunum, ileum, this pancreatic juice contain pancreatic amylase, and this amylase is responsible for the digestion of carbohydrate till carbohydrate become monosaccharide glucose. So this amylase work in mouth and in ileum, not in mouth and pancreas, not in stomach and pancreas, not in stomach and ileum. Basically the pH of the stomach is acidic due to the presence of HCl, pH acidic, and amylase denature. So there will be no digestion of carbohydrate in stomach. Only protein digestion will occur in stomach. Which substances are transported in xylem and phloem? Xylem is associated with the transfer of water and minerals from root to shoot, and phloem is responsible to transfer sucrose and water. So this is the right options. Option number C is the right option. Get mineral iron and water, sucrose and water. So in next video we will solve O-level biology past papers of year 2010 paper 1 variant 1 MCQs number 10 till 20, and this should be the part 2 of this video. Thanks for joining us. Thanks for following us.